Hello, everyone, and welcome to today's webinar, Multidimensional Approach to Decoding the Mysteries of Animal Development. I am Matt Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by GenScript. To learn more, visit genscript.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Justin Crocker, Group Leader, EMBL. Justin, you may now begin your presentation.
Hello, my name is Justin Crocker, and today I'll be walking you through, kind of really what's at the heart of a lot of what we research within our group and what a lot of the field is really motivated by to really explore how genes are expressed across development and evolution and how changes to these can actually result in, a variety of diseases as well as beneficial things like changes in organisms that contribute to very novel features. So what I'm showing you here on this particular slide is really what the core of our research, which is fruit flies, right? So really what's important here, and I'm going to walk you through a very deep dive, why we research something like fruit flies, why we think it's a very powerful model system to really explore much of the life and the biology around us. Because what I'm showing you here in the background, these simple A, T's, G's, and C's, these nucleotides that make up the DNA that actually drives the gene expression patterns of what you see here, which is a developing embryo in this magenta, because it's actually a ball of cells in a very, very early embryo of a fruit fly. And what you can see is that these A, T's, G's, and C's, which seem very simple, are sufficient to actually drive these beautiful patterns of expression we see during development, right? So this is really something that's been at the heart for many researchers in the field for a long time. I'm going to walk you through some of this history. And again, because what we're really interested in within our group is using this as a model system now to connect how these A, T, C's, and C's give rise to these patterns of expression, and ultimately these adult forms, in this case of a fruit fly, but this could really be any particular system, right? So how do you actually drive these patterns of expression, and how do they manifest, in this case, as a fruit fly? So really at the heart of our research, again, is something known as transcriptional enhancers. And what I want to walk through first now is, kind of the focus of why are we interested in transcriptional enhancers? Really, what are they? What are they doing? And how within our group are we studying these? And then I'm going to do a real deep dive and kind of, walking you through the actual experiments we're doing within our group and the new biology we think this is revealing, right? So first, what are transcriptional enhancers? In a very simple way, as you can see here in this diagram, transcriptional enhancers are these little regions of DNA, and what you see in these long arrows then, these arrows are actually kind of these interaction diagrams, if you will, right? So these enhancers, what they're actually doing is these little developmental switches that will actually tell this particular product, in this case known as pol2, which it's not really important, but really what this molecule is doing is turning a particular gene on, right? So these are structural genes, right? So this could be actually, in this case, different enhancers. These switches could be telling a particular gene to turn on, for instance, in your eye or a limb or a leg, right? So again, what these are actually doing now is, in this case, for instance, tissue, the enhancer A could be turning on in this particular tissue type. So if it's in a vertebrate, it might be the developing limbs. If it's in one of the fruit fly embryos, which we're studying within our group, it might be a particular stripe. And then in some cases, you may have a different particular one that's turning on a different tissue type, right? In this case, tissue B from enhancer, right? The switch might be turning it on in a particular region now in the brain of a developing mouse, or it could also be different stripes in the embryo, right? So again, the important point here is that these little developmental switches, if you will, they're turning these on. And what we're really interested in understanding is, if we have these stretches of DNA, can we actually predict where these enhancers might be? Right? This is the interest of many, many groups in the field. And then if we have mutations to these regions, are they actually going to change these different stripes or change where you're driving expression during the brain? And the reason, again, we care about these particular things again, right? Just to reiterate this, 'cause it's very important throughout the talk. Again, these are little switches that turn genes off and on during development, or just normal tissue physiology. And they'll do this actually by, in this case, I have these little things labeled as TFs. What that means, that's this shortcut for transcription factors, because they're actually sitting there and they'll bind to these switches and they'll tell it to turn on or off in particular tissue types. That's really functionally how they're working. But there's a lot more nuances to that, as we'll get to throughout the course of this talk. And what I also really, really want to emphasize is that we often think about genes, as these simple little protein coding things, like structural things, again, like hair or eyes or whatever components there. But the bulk of our genome, right, if we look at all these letters in our genomes, right, so these millions and millions of base pairs, most of these are actually these developmental switches. And yet, as I'll walk you through our talk today, we still have an enormous amount to learn about these. So again, why do we care, right? So what functionally are these doing outside of just turning things off and on during development? What I'm walking you through now is really an examples of how enhancers are actually driving evolution, and which is at the core of much of what we work on. But a counterpoint to that is when things actually go wrong, which is actually disease. So first, some nice examples of evolution. There's examples such as the prefrontal cortex, for instance, in vertebrates, right? So a big innovation that happened as we went from mice to chimps and humans, these different phylogenies, is that the particular regions of our brain got much larger. And a lot of this is actually thought to be driven by these switches during development, which is actually encouraging extra growth of the brain, right? Linked to different things like emotions and intelligence, right? A lot of things that are thought to be rather unique to humans in this case. There's other very unique things, such as in this case of eutherians, which is another very particular thing in this case for humans, which is placenta, right? So these are entirely new tissue types, and again, these are thought to be driven and arise throughout the process of what's making us uniquely human through, again, the gain of different developmental switches, right? So again, these are very, very important. And there's many, many other examples across evolution, such as in these beautiful pigmentation patterns of fruit flies, which again is what we study. We have different things such as fish, which is the armor plates, right? So these actually change between species. So again, each of these are very, very nice examples now of how changes in these switches are really driving a lot of the biology and I think the beauty of life around us. And then I think these are other very just, kind of dramatic examples, for instance, that the limb evolution in snakes, right? Snakes actually lost limbs at one point during development. This, again, is a change in a developmental enhancer, right? Also things such as the growth of bat wings, right? All these beautiful different features. And also to come back again to disease or different changes or pathologies that might manifest during things that happen in these different systems, these are also now linked, in this case, to over 90% of human disease are associated with particular regions of these developmental switches, right? So if you change these, very bad things can happen in some cases, but in the evolutionary perspective, you may actually have good things that'll happen. And we'll kind of continue throughout this during the course of the talk today. So really at the heart of a lot of what we study and many, many groups is, again, this very, very simple relationship on the surface, which is how these ATs, Gs, and Cs, right, which I keep kind of harping upon. But what I'm showing you here is that we actually take this little strip of DNA, and we put this in front of a reporter gene, as we call it. In this case, these could be things that like GFP, green fluorescent protein, or other molecules that actually light up during the embryo. Like I'm showing you here, this is the early Drosophila embryo. And you can see that what we actually have on the left-hand side in this case will actually be the developing head. And then the other side will be the tail. And you actually have the belly facing up. And what you can see is these beautiful, beautiful stripes that will arise over and over again, very reproducibly, simply by this little switch, right? So this is actually the readout of where the switch is turning on a particular gene. And so what we're interested, again, in understanding now kind of this textbook example, which I'm showing you here, because literally if you look up, from textbooks and all of that, you have this very simple diagrams where you might have, a few these little molecules, these transcription factors that'll bind to these particular regions of DNA. And in turn, they'll tell it to turn on in a particular region or off in others, right? So it seems rather simple on the surface, right? You just have these little sites maybe you can look for in the DNA. which might predict where it'll turn on. But what I'm going to assert right now, and I'm going to show you many examples of this throughout the talk, is that we cannot build these by very first principles, right? In other words, if I just have these collection of binding sites for these transcription factors, we put these in front of a reporter gene, we typically have nothing that I'm showing you here that's really reproducible, right? So by first principles, we still have a lot to learn as a community. And another major thing is that we really struggle now as a community to predict where these may be, right? So many of us could actually go out right now and have our genome sequenced. And yet, if we have just this very simplistic sequence, we can't just look at it and say, you know, this is a roadmap for building a system, right? We still have an enormous amount to learn as a community. And this, again, is really what the emphasis of both my group and many others around us who are in my host institution, which is here at the EMBL in Heidelberg, right? So really what we're focused on is understanding what do we not understand about these, like, you know, these little developmental switches. Why can we not predict their activity and why can we not build them? So really, you know, fundamentally, it's just what information are we missing? And these are very simple things I'm going to walk you through using this as a kind of a case study. You know, for instance, how many of these binding sites for these transcription factors will sit down and tell it to turn on during development? You know, is it 1? Is it 2? Is it way more than that? There's other things, just, you know, how robust are these? If we start mutating these, right? Because if I already told you that these are at least linked to different diseases, you know, if we actually go into these and mutate these, is it predictive, and how often might it cause different changes in disease? And then, you know, how do they contribute to evolution and the different morphologies that we see around us? And then ultimately, again, what many of us are trying to do is now, if we do have this genome, if our genomes are sequenced, how predictive is it for, you know, changes that might manifest as different pathologies? So the reason I work on something which might seem on the surface rather ridiculous to many people, which is fruit flies, is I'm going to kind of walk you through this here. And the system that we actually use has an even more ridiculous name in this case, which is known as Shaven Baby, right? And the reason we have this very crazy name, and this is something that actually kind of has a history to it in the fly, is that what I showed you earlier, these little cells that are turning on during development, these little strips here, these actually link back to this very classical work, which was done right here in this, right down the hall actually, by Eric Wieschaus and Janne Nielsen Volhard. And what they actually did was a screen that went on to actually win the Nobel Prize here. And the reason why they won the Nobel Prize is they were working in the very same system I'm going to walk you through now. And by doing this, if you see these little kind of wiring diagrams above that embryo I showed you before, is these are all these very classic signaling networks, right? So what they were starting to do is if you mutate different parts of this, they were just doing these random huge mutagenesis screens and seeing how it affected the output of these particular stripes I'm showing you here. And classically what they did is actually looked at the larvae. So very after this stage of development, I'm showing you the nice magenta here. If you see underneath this nature cover here, this is the work that again led to this Nobel Prize, because what they were looking at now is these weird little segments in the larvae, which we know as denticle bands. And really what these are doing, these are little hook-like structures that allow the larvae to crawl along in food, right? So any of you had fruit flies in the kitchen, you know, pretty horrible things can happen depending on your perspective. Whereas if you look in, for instance, your trash or fruit as they're rotting, you know, you have these gross, disgusting maggots crawling around. And the reason they're crawling is all linked back to this particular gene that these groups of many others have now been studying for many years, known as Shaven Baby. Because again, these are little hook structures that allow them to just crawl along. So again, the important point here then, after all that kind of lead in, is that we have these 8 different denticle bands again, right? These hook structures, and these directly correspond to these different aspects of expression you can see here labeled 1 through 8, right? These are the same little segments that we see during the embryo. So the important point here now is we have decades and decades of work, many different groups now who have really been focused on understanding how all these classic signaling networks, right? And again, I want to emphasize these same networks exist in humans and pretty much everything else we see in nature, right? These are very deeply conserved networks. And it's much more developmentally how you kind of use these networks. And in this case, how these networks all are integrated at the level of these little simple transcriptional systems. What I'm going to walk you through now is we don't actually think these are very simple at all. And this has been a real challenge for the field. So the kind of unique viewpoint we took to kind of explore this space now. And again, how these very simple enhancers from the textbook viewpoint might work was to actually do a very, very deep dive here and to really explore in a very unbiased way how this information may be distributed across the enhancer. And really key to that was the generation of these enhancer libraries, right? And so what we took is that same 300 base pair enhancer, and we just did a random 2% mutation rate through this, right? So again, it's a very stochastic process. You kind of have a distribution where some have 1 mutation, some might have 10 mutations. And by doing this systematically over and over again, we had about 1,000 independent Drosophila lines where we put every single one of these in the same reporter gene, every single one back in the same spot of the genome, right? So it's super clean way to go through and precisely measure how individual changes in this switch might relate to changes across development. And again, I really want to thank the sponsors for this GenScript. As you'll see throughout the talk, they're really kind of the people in the providing much of the infrastructure for this. And they were the first people a long time ago, which, I mean, this is a very, very long project for us, as you'll see, which started maybe a decade ago now, where they've really been partners who are enabling us to build these libraries. And what I wanted to then do once we have these libraries, again, to make 1,000 lines, which I won't go into now, but always, again, feel free to reach out to me after the talk. I'm always very, very keen and very happy to discuss these much further, because there are a lot of ways now where we can put these back into the fly in the same exact spot, you know, really at scale, so we can make thousands of lines. And the next step after building these thousands of lines is we actually had to have ways to really handle many, many, many embryos and really to do this at scale very systematically. So really at the heart of this, which I'll walk you through very briefly, is kind of this automation system, these robotics to actually handle all the embryos for us to, again, see where they're being expressed during development. And I'll go a little further into this in a bit. Because part of this also now is really, by doing this scale, we really wanted to treat this much more as, if you will, a physicist would, right? Approach this very deeply, very systematically. So again, now we can relate very simple changes on the surface to single mutations, for instance, in the enhancer, to these differences in output. In this case, what we mean by output is if you see underneath this imaging quantification thing, all these little tiny little dots there, each one of these are actually a single embryo on a slide. And this is actually an enormous, enormous amount of data, which again, I'll continue to walk you through really to kind of impress upon you really the power of scale and really what this is starting to buy us as a community going forward. So really at the heart of this initial body of work was a lot of this robotics here, and there's a lot of excellent collaborators. My previous advisor, David Stern at Genalia, and many other super talented engineers who helped us to really, you know, enable a lot of this technology. Because what this allowed us to do is to take, you know, these fairly simplistic fruit flies, we could actually put each individual line, right? So every enhancer variant into a cage, and you can see these little wells. If you zoom in on their individual wells, you can see it underneath this little embryo heading. Each one of those weird looking things there is actually an individual embryo, right? So you get lots and lots and lots of these. And what's super fun for us now is we can actually take these little wells, drop into the robotics here, just hit go, walk away, and it does all the staining and fixing and all this other stuff, which previously was not just laborious, but it actually, you know, these are really dirty, dirty assays in the sense that if you're going to do 1,000 of these, your ability as a scientist to, you know, handle these appropriately, adding all the chemicals appropriately is actually very limited. And robotics actually afford us to move past that point. And following a lot of the imaging or the staining, the next really key point then was actually to assay these through the imaging. And really at the heart of what I'll walk you through here, now we have the ability through the robotics to actually, you know, we take these, we put multiple wells down to image these really at scale. And if you can see as you go through this diagram here, through the automated imaging and registration, again, we can just take these slides now. We can put a whole bunch down at once. We can do these overview images. You can see here, again, each of these little things that look like a grain of rice in this case. In this case, these are actually each individual embryos. And what I'm going to try to play here is a video where you can see that as you zoom in now, we can automatically go through and take many, many, many pictures of these. And I'm going to show you here now in green. This is actually the full shaven baby expression pattern. And in the magenta, again, this is actually will be where if you see the reporter gene, the wild type one, if you put it into the embryo again, where it will be expressed in relationship to the full expression pattern. So what I hope you can see now as you rotate through this is actually a full 3D confocal projection now, right? So these are very, very rich, very deep, powerful data sets for us to, again, to query how you have changes in enhancers might relate to changes, in this case, of these aspects of expression during development. And now what I'm showing you, which I think you'll see this video playing, and it's going to be a kind of insane amount of data coming at you really fast. And that's kind of almost by design. It's kind of like a shock and awe slide, because one of the most dramatic things we found by screening all of these 1,000 lines, and I hope you can see this in this video as it goes really, really fast here, is that every single line that we tested have a different aspect of expression, right? So this was, to us, very, very jarring at the time and totally, totally unexpected. Right? So again, what this high throughput embryology bought us, right? And you can get some of this as the references here. So I'd be more than happy to discuss that scale. But really what this allowed us to do now is to really, again, explore this enhancer in an unbiased way. And really, again, the very dramatic thing, the reason we think these are so hard to predict and understand is that every single line we tested had a different aspect of expression. And I think outside of kind of these insane videos that go super fast, right? Again, you can see now that there's a huge amount of difference of this expression. But now if you actually slow this down and then really look at all this breadth of data, and what I have here now, if you compare to wild type, you can see that it has in the kind of middle band of these embryos, wild type has this very nice stripes again of expression, these very reproducible stripes, embryo to embryo. But now if you go through all these different lines, each of these ones with little numbers next to them, these are actually single variants. So a single nucleotide is changed in these. And what you can see that, again, is very dramatic to us is that every single one of these had a statistically different level of expression. And by using all this different information, I'm not going to go through all this now because this is published, but what on the top I'm showing you now is what we call a footprint score. We're able to actually go through now and see regions of the DNA. And in this case, you can see underneath it, many of these are actually associated with known motifs. But in others, you can see that these footprint scores, really what this is telling us is the probability that if we mutate something here, you actually have a very, like a collapse of expression, right? So again, there's a huge amount of information through this, at least from this mutational perspective, right? This was really, for us, was totally unexpected. And this is why it goes on to be kind of one of these fancy journals here. So what I want to walk you through now is kind of just one little simple case study to show, again, the power of these type of assays by combining these like synthetic approaches with all this DNA that GenScript provided, putting it all into these systems and really exploring this space in a very deep way. Because what you can do now, for instance, here you can zoom in on this one kind of motif that at the time we had not explored further. I'm going to walk you through now how we can actually footprint this and how we think this is giving rise, as we mutate single nucleotides again in this case, will actually collapse its expression patterns. So by zooming into one of these now, you can see in the top here, this is what we're calling coverage. This is if you just take this entire library, align all these different sequences, and you can see that these are just mutations per site. So you can see that we have a lot of coverage, and across a library, we hit every one of these different particular nucleotides across this developmental enhancer. And now you can do a very simple thing, just say that, if we go to our library, just pull out different random lines, in this case, that have at least a C mutation in one of these nucleotides. In some cases, they have others that are distributed out. But what I hope you see, if you look on that axis here, I'm showing you the activity, right? So the log change of different lines that have a C mutation in different spots as enhancer. And what's still, I think, pretty remarkable is you can see that there's certain regions that you can almost footprint, right? Shown in this case by these little red bars that go down, because if you mutate these to a C in this case, you will have a very strong decrease of expression. And now what I'm showing you also in these little highlighted regions, one of these here is shown as in kind of this green box and the other in magenta. The reason we've highlighted these in the green box is this factor, which really isn't that important for this particular talk today, but if you're a fly aficionado in the audience, this is one known as a Hox gene for ultrabithorax, which is another very classic fly gene. But again, the important thing is that we knew this was a binding site for a transcriptional activator. So this is kind of our positive control. But we also had these kind of new motifs that popped up throughout this type of assay, known as these HTH or homothorax motifs. So what was quite nice is that we can actually do now, go in, specifically mutate, in this case, this UBX1, this in this case is positive control. And we knew that if by mutating this, you can see that if you compare the wild type embryo to when we delete this particular motif, you can see that you get this really dramatic just kind of crash out of expression. But prior to this, we had no idea what these other homothorax motifs, which we now know they're homothorax motifs. But prior to that, you can see that you can actually footprint these type of regions. And now you can see that if you do the same type of thing by mutating these in the reporter gene, putting it back in the fly and comparing it to wild type, again, you can see that similar to what we're getting from these libraries, you get these super strong decreases of expression by mutating this. Again, so we think this allows us to footprint these different binding sites. And we can also do different things like show how they're cooperative, which I won't go into all the details now. But again, by mutating both of these particular sites now, we can see that you have, in this case, almost no expression. Actually, there's a single little nucleotide if you kind of zoom in there on this one, right? So maybe you have a single, sorry, not nucleotide, a cell in the entire embryo now that is showing expression when you delete these, right? So clearly you can really identify, you can footprint sites. I won't go into all details. I'd like to kind of just show this because there's always some kind of like old guard biochemist who wants to, validate all these. So we can do this. And I really have to thank Richard Mann and at the time, this very talented postdoc in the group, Nomiko Abe, right? Because they did this. These are really, really hard because they're very low affinity binding sites. They're non-canonical sites, but they clearly, these transcription factors will bind to them. You mutate these and you no longer have this binding activity. And furthermore, what's quite interesting for us is that they'll physically cooperate. So you get these like super binding events by having both of these sites. So again, I won't talk about this now, but if you want to go offline, ask questions about any of this, I'd be more than happy to address this. Okay, so at this point, we've kind of gone into what we thought on the surface was a pretty simple little piece of DNA, right? This little switch. But now we know that these are very, very dense. There's an enormous amount of information that we really did not expect at the time. And, as I alluded to, but I didn't go into that deeply, we think that one of the reasons they're so sensitive if you change individual nucleotides is that there's a lot of cooperativity here to make this like kind of a potent switch, right? Meaning that you have different molecules interacting with one another biophysically. And now if you disrupt this by, either the interfaces or where they're binding a DNA, you just have this dramatic collapse of expression from these particular regions. So really, this kind of started us down this road to understand, if they're this sensitive, how evolvable are they? And again, because a lot of this relates to different things like disease and many other conditions, right? So how can we relate these changes again now in more of a predictive context, right? It's kind of one of the goals again for both our group and many, many others in the field. And before I go too deeply into this, I thought it'd be a nice kind of pause inflection point to go back to really one of the, people who started the entire field of evolution, in this case, Darwin himself, right? So the concept I'm going to walk you through briefly here is something for developmental biologists, in particular, Evo-Devo people, or evolutionary and developmental biologists. And this concept that Darwin at the time, I would argue, was talking about developmental constraints when he talked about something called unity of type, right? So this is a very old concept where, and I'm going to read you this quote because I think this is kind of, this is very contentious in the field. People like to fight about these things, particularly evolutionary biologists. So one of the current definitions, and I'll kind of punt this to them, but again, I love to discuss this, so please get in touch with me after if you'd like to, further fight these different viewpoints, is that developmental constraints are a property of a trait that, although possible in the environment in which it originally evolved, acts to place limits on the production of new phenotypic variants. And the reason I'm showing Darwin here is he noticed this relationship between the size of the beak when he was looking at his classical finches, right? So where it would actually interface with the skull and then how long that beak was, right? So it seemed this very simple relationship that, which I think in hindsight makes sense, where if you have a longer beak, you need more support in the brain, kind of the brain case where it's interfacing the skull. And this has actually been formalized a bit now. And this is one of the classic signatures of one of these developmental constraints from the perspective of, us as evolutionary developmental biologists, in that you have, again, these kind of linked traits where now if you actually extrapolate these type of principles, in this case, I think it was buzzards actually, not finches in this case. But now you can see that across many of these, you have this very tight linked relationship, right? Because they're kind of moving together in kind of this morphological space, if you will. right? So again, while this has been described for a long time, there's many ecologists, right? Many people go out in the field, collect specimens, make a lot of measurements. You tend to see these relationships all over the place. But really, there is very little molecular insight or molecular understanding of the underpinnings of, why we see these types of trends and these traits in evolution. So what we were interested now, because of what we're starting to learn about these enhancers, is do we see these type of things, or in other words, does enhancer logic place limits on the production due phenotypic variance? Do we see these type of trends in these linked relationships? So one of the things I'm going to walk you through is one of the first kind of compelling examples of this, whereas if we looked at wild type enhancers, we knew from some of our previous work, if you tuned up these elements by increasing the affinity, right? In other words, how strongly one of these transcription factors could bind and activate the expression of something, that quite often what we would get is something that it wasn't just turning on in higher levels where it's normally expressed, but I hope you can see that in the, kind of the anterior region of these embryos and many of the side cells, right? This is that same factor I walk you through, the kind of the biochemical validation I alluded to very quickly for this factor known as UBX, but this is not normally expressed all throughout the entire embryo, right? So what's happening is by increasing the affinity, you start to have activation, not only of that transcription factor, but all sorts of things will start clicking onto it and turning it on inappropriately, right? So again, this is something that you do not want to have happen, because very bad things can happen, because this particular gene, remember, is actually turning these particular cells into these hook structures. And functionally what this means is these cells actually die, right? Because they'll become these little morphological hooks, where most of it's just faded now for cuticle, right? Something much like our fingernails, right? So you don't want to misexpress this in the wrong location, because clearly it's going to have a very bad effect. And not only do we see if we turn it on in these higher levels by increasing the affinity in the wrong place, we also turn on inappropriately early, right? So this again is another thing that we see, this kind of relationship now where you have many, many bad things happening across developmental in terms of the time, the spaces, and locations here. So I'm not going to go into this too deeply, but again, this is for people who really are kind of committed to going down this kind of quantitative route like ourselves, where what you can actually do now is go in the earlier embryos, right? So a stage earlier we're normally expressed. And what I have here in the bottom here, we can actually zoom into these anterior or the intra or interstripes, right? So either kind of, we call these naked regions of the embryo where you don't normally have expression, or you're normally showing expression. And what you can see at these early stage 14 embryos, the higher affinity one is already starting to turn on in that stripe, right? So we label this as a stripe at the stage 14, right? So this is actually the stage prior to what it's normally turning on for Drosophil development. And this becomes even more exaggerated now, not just in where it's normally expressed, but you can start seeing that in between stripes now. If you quantify this, you see this is turning on all over the place, right? So clearly this could be quite bad. And then to take this a step further, what I'm showing you here in this kind of 3D plot to kind of break this down, what we have now is the intensity kind of going up and down. The size of these circles actually relates to the affinity, right? So what I'm showing you again at the top, just to remind you then, if you have the wild type, you increase the affinity, you start turning on it appropriately. And that big, huge box is actually kind of the extreme example I'm showing you in the top. But all the others, you decrease the size of the box, what you start to see again is this very kind of nicely linked relationship that I alluded to earlier, right? Because now you see that if you go from wild type, which actually this teeny tiny, very, very weak binding site that we think gives us an enormous amount of specificity in this case. So it's uniquely binding only this one transcription factor, and there's thousands of these typically that could be expressed in any given cell, right? So there's many transcription factors, but by having low affinity, we think this is unique for this particular one. But now as you increase that affinity, you increase the size of those, in this case, these circles that are representing that, you can see that you'll start turning on earlier in stage 14 for all of these. This is also associated with ectopic expression outside these other zones, right? So again, you have this very nice linked relationship. And we think that, I won't go into this again now, but we think that as you go for specificity versus affinity, as you increase this affinity, kind of this tuning that I'm showing you here, what on the top this actually starts to do is that these other transcription factors actually start to compete for these different sites, so such that things that normally would not bind them will stick there, and they're turning a switch on all over the place in the wrong locations. This is why you get these kind of crazy expression patterns. And so one of the other things we were quite obsessed with at the time is to see, how far could we push this, not just from these transcriptional activators, right? Because you can imagine it's kind of easy to make something stronger or weaker. But one of the things that we knew going all the way back to that original screen, this classic Heidelberg screen, if you will, is that you have these really interesting mutants, in this case known as wingless. Again, the identity is not really that important, but this is like TCF for some of these other ones for more vertebrate systems. But what's interesting about this is that we know now there's a transcriptional repressor. And this is what we learned about this, and one of the reasons we first identified this, or the others in the community did, not myself. But if you mutate this gene product, you, instead of having this nice, clean little hook structures, right? And I hope you can kind of see that here, where you normally have these naked regions. Now, if you remove this product, you can actually get a lawn of these. And it turns out what this is doing again is it's making these naked patches by actually functioning as a repressor. And you can see that, I hope, in this diagram here, these little hook-like structures, these are actually the kind of this cartoon version where normally this enhancer is the magenta here. These are each little individual cells that are represented in this diagram here, where some actually have the cuticles for shaven baby. The reason it's not expressed everywhere is you have, again, this morphogen gradient, or this areas of expression which are going to signal actively to turn it off. And in the enhancer, what we noted, and I hope you can kind of see this now, that the motifs for that are effectors for this are actually oftentimes overlapping directly with some of the transcriptional activators we found, right? So they're kind of competing for a site. So we were curious about, the consequences of this, because we, in our collection, we never had any examples of this big lawn of cells, right? And we were curious why we were not getting that. So we spent a lot of time looking at this, and one of the major things we found is that every time you would touch one of these binding sites for this transcriptional repressor, because it's directly overlapping one of these activator sites, they're always coupled. Meaning that, I hope you can see on the right, this line known as E3N97-3 is one of these variants in the library. So now we have a single mutation of the repressor binding site, but by kind of augmenting the directly vicinity of this homothorax, that other site that I talked about earlier, right? So we knew that if you mutated this site, you collapse expression, but even mutating something in neighborhood will also collapse the expression, right? So I hope you can see in this particular line now that where your normal express goes down, you start turning on a little bit in between cells, but it's coupled always with the normal expression going down. And we can actually go through and quantify this, where again, the wild type now at the very high levels by this bar graph, this is actually where it's normally being expressed. And next to it now, the very low one, this is the wild type one showing the expression in between the segments, this naked region. And you can see it's normally very, very much off in this naked region. And we can actually extrapolate this further now, really showing that across many of these lines, we have the same thing that we found for this particular embryo on the top, where now if we mutate this repressive zone, you always couple this with a little bit of ectopic expression in between, but it always comes at the cost of your normal expression zone really dropping, right? So again, these very, very linked relationships. We see this across many, many different variants, right? We can just keep doing this ad nauseum, if you will, because again, the really kind of potent thing we found here again is these linked relationships now, where the reason we couldn't get that lawn of expression is because every single time we started mutating this, the normal expression would decrease, right? Such that, we could never get expression throughout the entire embryo. And then we went actually further and said, okay, now if we think we understand the system by doing this really at scale, if we go out into the wild and look at many, many, many of these variants, what do we see? Right? Because the prediction was we should never actually see a lawn of trichomes ever in nature if this truly is manifesting now as a constraint. Right? Again, this very linked relationship. And so we actually did this. We went out and gathered many of these, or like over 50 variants. And a lot of this was David Stern and others who got many of these lines together. So we had millions and millions of years of evolution. And really the striking thing is that we found is very consistent with that very simple hypothesis. Based on this constraint, we think we never ever had expression in these zones, right? And here's a few different examples. On the top is melanogaster, the one we work with as the common fruit fly. But many of these others, you can go from the desert in the US or different regions throughout the world, we never had expression there. So again, we think that, going back to Jacob and many others, that, his particular quote was that these are adaptively coherent patterns. And we think that by doing this at scale, as we walk through this example here, we hope to identify many of these patterns. So at this point, I hope I've kind of convinced you that, this is a textbook example of enhancers, but we think that these are incredibly information dense. So it's much more something like this, right? It's just blanketed with information. Many of these are probably transcription factor binding sites we haven't identified. There might be other structural elements to it in terms of the rigidity of DNA, a variety of other things. So again, there's a lot of information here where we think as you mutate it, this is having a dramatic effect. And we were kind of interested in exploring further how common are these constraints, because I think, from many different perspectives, it can teach us a lot of biology. So I'm going to walk you through now a little bit about looking at other tissues, other enhancers before we go to, kind of wrap up the talk. And I'll go through this a little bit fast, just in the interest of time. But again, always ping us because some of this is already out in preprints. But one of the things we noted in terms of constraints is, again, we focused a lot on these kind of nice patterns here of this one known as shaven baby, which again, now I'm showing you here in green. But what we found is across our entire collection, not only did we never see it, in between these stripes as I walked you through, we also realized that we only saw it in particular cell types where shaven baby was normally expressed, right? So you have these different variants that I'm kind of showing you examples of, different posterior, kind of the bum of the embryo, with some that would actually be developing wings that would, go on to be wings later on. But the important point here is that these are all patterns of expression in native zones where shaven baby is normally expressed, right? So something fundamentally important we think is happening here. And you can actually quantify this in this 3D plot here. We just kind of parse it apart in kind of these classic tissue zones, right? These germ layers, if you will. What we found is that as you mutate all of these different variants, which I'm showing you here, kind of going up and down, every single one of these was limited solely to this particular ectodermal tissue zone, right? We never had anything in the mesoderm or the endoderm or any of the other classical tissue types, right? So in this case, it was kind of shocking to us because, I kind of grew up intellectually thinking that enhancers were kind of, simple switches you can turn on and get easy expression anywhere. But at least from these standing elements, the ones that are in the genome doing something important already, by mutating them, it's kind of biased already. Right? So again, we were curious if this is general. So I'm going to go you through this a little bit quickly, again, in the interest of time. But this goes back to this very classic concept in developmental biology, where shaven baby is known at the kind of this differentiation cascade. This goes back to kind of Waddington, and many classic biologists have been kind of building upon this, where if you have this little marble in this trough, kind of this metaphor is that as you go down, your marble is going to kind of start differentiating, and then it gets kind of locked in. So it could be that shaven baby, again, is this terminal selector gene, which is going to fate the cell to its final form, that maybe it's so sensitive because it's some kind of, way the switches are set up for something that is very late in development. So maybe if you go earlier, in this case, it's mid or early in the embryo, and I'm going to walk you through these examples now to see if we see the same type of patterns, right? Because maybe they're less hardwired, they're a little more plastic, and they can move around. So this is another classic Drosophila enhancer known as a Tin Man enhancer. And you can see that this kind of beautiful expression patterns now in kind of this yellow diagram. So these are the zones where, again, this reporter gene drives expression in developing muscle types. And we think that we know quite a bit about this, many of these binding sites. But what we found, again, going through this, through these similar enhancer libraries, is that if we mutated this, there was one, huge amounts of change of expression, right? So many, many sites outside these canonical motifs that were previously explored. But again, everything was limited to this one tissue type, only in the mesoderm, right? So this, we think, is still fundamentally very important. And we, again, just never saw it turning on in like the nervous system or any other tissue type, right? So this is pretty hardwired in this system. We've actually explored this to many other enhancers, such as this very early embryo one that is like one of the first times the mesoderm is actually, or the developing muscle will be marked. You can see here in this blue pattern. And on the right is kind of a schematic, because at this point, it's actually, the embryo is the ball of cells. This is like as early as it gets, pretty much, when their fates are really being assigned. So this is actually showing a cross-section of this. So twist is this particular gene, and this particular enhancer, the twist PE element, not really important, but this is the switch, it turns it on. And another one of these is just kind of these stripes on the embryo, right? So we have these different ones. The stripe will actually be turning on developing nervous system. And what's important for us is we have these, again, classic enhancers. We think we understand these binding sites, but consistent with everything I walked you through before now, if we build these libraries, test these enhancers in vivo, what we found now is as we mutate these, when we actually now assay the levels of expression in different zones, where I'm marking it here as the ectoderm, mesoderm is endoderm, kind of the proxies to see, how mutable and what kind of space can these enhancers move around in. What we found which I still think is quite striking, is that, again, twist was only limited to the mesoderm where it's normally expressed. We didn't get any extra expression. In this case, same with this other enhancer, which is in these nice stripes, which is developing nervous system for this one known as rhomboid. But again, everything was limited to these normal patterns of expression, which to us was quite shocking because I think there's an enormous amount for us to still learn here. And I won't go into this any further in this case in the interest of time, but what I can also impart upon you is that it's not like it's impossible, because we can actually add in a lot of synthetic activation here and kind of boost it all the way up. But what we found at this point now is that actually, even through this, it's possible to turn on these zones with the most potent, one of the most potent activators we know. But it's still really, really difficult as you go from, we can go for 1 or 2 or 3 binding sites that we add in onto this enhancer and just ubiquitously express the activator everywhere in the embryo. And even here, it's very difficult to turn on these inappropriate tissue types. Right, so clearly, this is another classic enhancer. I forgot to put this in here, but I'll go through this in the interest of time again. This is literally a textbook enhancer. So if anybody's taken a developmental biology class, you've learned about something known as even skip stripe 2 enhancer. Literally, you can go look at, I have this textbook sitting up here next to me, the molecular biology of the cell. All the identity is not important, but again, decades of work. We think we understand the binding sites. And when I'm marking this little cartoon of the schematic for the binding sites next to this enhancer, which gives us beautiful, beautiful, very precise stripe through what you can see this number of signaling pathways from this textbook. What we know now is that just like every other enhancer, as you mutate this randomly with single mutants, a lot of it just collapses, right? So there's a huge amount here. And we think that these are really functionally constrained, these elements. And so what we really became interested in now is what other forces from an evolutionary or a disease or any other perspective might be shaping and driving different expression. If you can't really mutate these, where does it come from? So for this, again, GenScript actually ended up providing us this very nice library of totally random sequence, right? So we could put this through the pipeline. So this sequence has never seen selection. It really doesn't have anything, impinging upon its fitness. So now if we test these totally random patterns of DNA, what was really shocking to us then is that unlike the things that were mutating, now if you just look at where they're driving expression during the embryo, these things really go crazy. And this, again, if you can see that kind of this pie chart right here, in this case, about 36% of these drove expression somewhere in development. And many of these examples here, these are each individual lines. You can see there are different spots. Some are turning on the developing intestine, the gut, the head, the ectoderm, all over the place, right? So there's very high robust expression. So we were kind of shocked about this. So we looked at these different pieces of DNA, because we, in this case, had many of them, right? Because this is what you can do with these type of libraries. And then we could pull out individual motifs in this case. The individual factors aren't really that important here, but you can see that every time it had some of these motifs, they actually had expression in particular tissues, right? So GATA-E is on the top. This is one that's known to be expressed in the gut. And every single time in the developing gut, you get this beautiful expression at very high levels. This is actually associated with this motif, right? So pretty much every single, or in this case, every single time. And there's another one known as grainy head or GRH here. The same thing, right? It's turning on the ectoderm. So we were interested then why these key motifs are associated with developmental expression. And in this case, what we ended up doing is that we had this hypothesis that there's these classes of factors that most of DNA is kind of very wrapped up in these little nucleosomes, so nothing might have access to it. So maybe what these particular motifs that are associated with active expression are doing is they're binding sites for these class of factors that have this pioneer factor activity, where they can bind, open up DNA, and then let everything else bind to it. And we think that this is potentially what's happening because you need these two different classes, one that's controlling the kind of the on-off or the state, and then the others are kind of the levels or dictating where you're being expressed, right? So we're curious then, very simply, do pioneer factors regulate these elements? And we bias this with 2 classic ones from Drosophila. Again, that grainy head that I alluded to, and another one is Zelda. The names aren't really important, but again, the important thing is that they're well-characterized pioneer factors. And what we could do then is now do the same libraries, in this case, bias it with a key motif. The Zelda is globally expressed now. So now we have a new library. We have a bias motif in the middle, sandwiched around totally random sequences. And now what's quite exciting about this, now we have a huge amount of expression. In this case, it goes up to about 88% of them are driving active expression. And you can see some very nice examples of this. So this is robust. It turns on like midway through embryogenesis. And what's really nice now is you get this beautiful range. It's turning on all over the place, right? Because we know it's expressed broadly very early and then gets refined in the ectoderm. So we think that, this really is a way these sequence spaces are being explored potentially through evolution. And we can go even further with this. Another example of this grainy head one. And this for us, and again, it's expressed in the ectoderm. So we made another bias library. And what's really powerful about this for us is you get 100%, which for us is a very rare number to hit in biology, which tends to be a little bit messy. But 100% of these actually drove very nice expression, kind of often these stripy patterns throughout the developing embryo, exclusive in the ectoderm in this case, where it's normally expressed. And then we're interested further saying, if we look in the genome, can we actually get evidence that they're actually driving this adaptive potential? And I won't go into the details of this, but the important thing is that we found these regions that are exclusively these UBX binding sites that we knew from CHIP were binding these regions at very high levels. And what was important is that these are exclusively found only in Drosophila melanogaster, so no other fruit flies that are closely related, right? So we think presumably they arose recently through de novo evolution. They kind of came up through like mutational processes. And what was quite striking is we tested these, they all drove expression. And furthermore, we think that these could be material for evolution now, because if we mutate these binding sites that arose kind of de novo, these pioneer sites, we see that you actually collapse expression in some of these key tissue types, right? So again, we think that what is happening is that clearly these new sites have this potential to drive expression. And we think going all the way back to kind of, the functional thing at the beginning of talk, why do we think these are important, is that what we found now is if you look at many different case studies, right? So these are often just, small sample size on like these massive things we've been building. But again, brain evolution, placenta, limbs, armor plates. Now we know that many existing elements, so limbs actually come from the breaking of one of these switches, right? You lose this element, and that's why you lose limbs. In fish, you lose these armor plates because you break one of these elements, like literally at a region of the genome that's kind of a hotspot for mutations. And then a counterpoint to that is many novel elements, in this case, oftentimes through transposons or de novo evolution, in particular like the placentas, transposons that are hopping in the genome. They're turning on genes at different tissue types. And we think that this is really driving a lot of adaptive potential. And also in the mammalian brain, right? We see that these regions are kind of these hot spots for rapid evolution, right? So we think these are all novel elements and they're often not standing variation, right? So we think that, again, these are very general principles we're starting to find by going at scale very systematically. And so I'm going to end most of this right now, just kind of at this very final viewpoint. I kind of want to assert this very, very quickly because, a lot of what we've been walking through is connecting genotypes to phenotypes throughout this talk, right? In this case, with reporter genes, right? So we can do a lot of robotics, imaging, automation at scale. We think this buys you a lot of statistical power. And, we can publish this. We can learn a lot of nice, cool things. A lot of the work I already walked you through, again, is in bioRxiv paper. So feel free to look at that. But what's been frustrating for us in much of the field now is we're still, because these are reporter genes, where we're still trying to struggle to how to connect this to how they might manifest as the adult, how in different environmental conditions they may change. So a lot of kind of what we're doing, kind of the call to the community right now, these what we call grand challenges in terms of connecting genotypes to phenotypes at scale, right? And we think this is one of the most challenging things to do as a community. And I'd love to keep taking questions further because, from the genotype perspective, this is very easy for us to solve now, right? Any one of us can get our genome sequenced. We can sequence any of these little critters out in the wild. But now connecting this in a meaningful way to things like, predictive gene expression, metabolism, cell or tissue types, and, going even further, how these manifest in external things like morphologies or behaviors, and then really how they, in different environments, how they're robust are they to temperature, to food choice, all the many different things. This is what we're really trying to go after. So a lot of what we're doing in the group, and I really, really want to emphasize, we love to discuss this. This is where I give these type of talks. Is that we're going for these very high dimensional data now to really explore this at scale. We call this developmental phenomics. We're now working with people like GenScript and others. We can actually go in and do very discrete synthetic manipulations, right, at the genome. We're doing things like CRISPR, many new techniques we're continuing to push within the group. So now we can actually start to do a lot of multiomics, every other kind of classic omic you can get your hands on, to really now give us this huge dimensional data sets to connect these different scales. This is what the goal we think of our group and where the field is really headed. And then what we can actually do uniquely in Drosophila is see how these will actually change using laboratory evolution over time. And again, I'm going to walk through this very quickly because I know we're kind of coming up to time. But a lot of what we're doing now to kind of relate this back to our talk, we're taking fruit flies now. We're doing, in this case, transcriptional enhancers, but we're setting this many other things to really see as we work with people like GenScript, can we build these synthetic libraries at scale? Can we do this now actually in the genome? Can we relate this, by using robotics and automation to do this at scale? Can we, image through this doing these different pipelines, because we walked you through these kind of nice examples earlier, right? So we can get a lot of data now. And really what we're starting to do in the group now is push us much, much further. And now connect us not to just a simple gene expression, but to other things that are intimately related like metabolism. So we're doing spatial metabolomics, and this is actually a larvae I'm showing you here. So now we can look at 100 metabolites across populations of larvae. We can do different things like single cell genomics. We can do animal behavior in the fly. And really what we're quite excited about, I hope you can see the animal behavior, right? Because these are little crawling larvae, as I alluded to in the very beginning and much of the talk, right? So there's a lot of data here. And now we're actually tapping back into ecology and evolution and see how these change over time. And with that, again, we think developmental phenomena will be very, very powerful to teach us how these genomes are working. And I would love to discuss this further. I'll stop here and thank an enormous amount of people who helped us. And again, we do have positions available because two of my first, fantastic postdocs just walked off to get their dream positions. So great for them. We're still kind of reeling, but, we're really on the hunt for other people who, kind of share our enthusiasm and our perspective to kind of take the science much further. And with that, I'll stop and take any questions.
Thank you, Justin, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. Let's get started, Justin. It looks like we have just one question here for you. Oh, we got two that came in. Let's see here. First question will be, you might mentioned this during your talk, but how did you determine the mutation rate for your enhancer library, and how important for enhancer library being unbiased?
Oh, it's a very good question. Yes. So for us, the mutation rate was actually between two closely related fly species at the time. So we thought it was a good kind of aim to hit towards something that was actually represents something we'd see in nature. And for us, the unbiased angle is actually very important because we wanted to explore all the possible kind of landscapes of these changes and how they contribute to possible change of gene expression.
Okay. And just one last question came in here for you. The question reads, if mutating the two HTH domains abrogates expression nearly entirely, what are other domains contributing?
Oh, very good question. So we didn't have time to go into too much of that, but there's actually specific cells you'll start losing expression in for different transcription factors or different signaling pathways. So some of them have very precise changes at spots within the embryo or timing-wise in the embryo.
Okay. Well, I appreciate it. I think that was all the questions that we had time for today. We wanted to say thank you again, Justin, for your time and your important research. We would also like to thank LabRoots and our sponsor, GenScript, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.